Okay, we've been spending some time talking about the evolution and expansion of gymnosperms. Let's spend a few minutes talking about the development of tissues to transport water. So in order for plants to grow tall, we need to be able to have a mechanism that takes water and allows it to be transported from the roots, where it's taken up from the soil, all the way to the top of the plant. How do plants do that? Plants don't have a circulatory system, they have a vascular system. Basically, it is a one-way pipe, and that one-way pipe system first emerged when we began to look at the lycophytes back in the Carboniferous. Here we have giant lycophytes that get to be 10, 20 or so meters tall. We have horsetail that might have been 5 to 10 meters tall. How is it that we can have plant life which is large? This happens because of a vascular system. That vascular system is a series of tubes that allow water to be conducted up through the interior part of the plant. Plants could not get large until there was a vascular system, because capillary actions will only allow plants to achieve a height of a couple of centimeters. That's why the bryophytes tend to be very short in stature. They don't have a vascular system. Water moves by capillary action. So what is this adaptation that allowed trees to grow so tall? Well, the first part is to create a structure that won't bend. That was the evolution of a compound called lignin, which is a complex polymer which imparts rigidity to cellulose. So what happens is that we have cellulose forming the cell walls, and those cell walls are made rigid by a compound called lignin. Okay? That's the first feature that was needed, something to keep the tissue rigid to defy gravity. But the second part was the vascular system in plants that allowed water to be moved up. Water is moved up through a plant as a tension. So it's a passive uptake, and water is under tension. That it is a metastable condition, but it's like taking strings and stretching them apart, where the elements are held together by hydrogen bonds. If this was class, normally, we would show this exercise by imagining that students sitting in the seats next to each other were water molecules. They are held together by hydrogen bonds. That's one student holding the hand of another student. As water evaporates at one end, it creates a tug or pull. That pull is then felt from one water molecule to the next, and so water is pulled up under this negative pressure or tension. If that tension becomes too great, then snap, just like pulling hands apart. We have a cavitation, and that cavitation is exactly like a blockage when a human or other animal system has a stroke, a block in the circulation system. So the water is under tension. It's passively moving through dead cells. So it's moving through essentially an open pipe system under a negative tension from the roots all the way to the top. How does this happen? Well, we have the cohesion, the connection of one water molecule to the next, just like two students holding hands, and we have adhesion. That is, the water molecules adhere to the sides of the cell wall. In this case, the cells are called xylem. They're dead, water-filled cells. So the transpiration stream pulls up these water molecules that are cohesively bound to each other through hydrogen bonds. They're carried up through the xylem, and the cell walls on the xylem are made thick and stiffened by the presence of lignin. Now, in order for water to reach a height greater than 2 to 3 centimeters, which is what would happen under capillary action, the evolution of a transport system was required. So the transition from mosses to the next evolutionary step, which was the ferns, was associated with the development of a water transport system. A simple water transport system would be a wall around which there is cellulose in a ring-like structure to provide some rigidity and cohesiveness. And at the end of each of these cells, there are perforations that allow the water to be pulled up through this cell and then up through the next cell through those perforations. So a vascular system moves water under tension, it's a negative pressure, with water cohesively bound to the cell walls. So simple water conducting systems, like you might think of in a moss, maybe in the sporophytic part of a moss, are simply a primary wall which is elongated, has very little structural support, and if water is in short supply, it's under more tension, these cell walls will collapse. So mosses are only able to maintain some sort of a simple capillary action only in the sporophyte if there is adequate water in the environment. The first vascular tissues that we can find fossil evidence of are in the Cooksonia. So here is a reconstruction of Cooksonia based on fossils. And when we begin to look at this in greater detail, what we find is evidence of a primary wall with additional rings associated with cellulose. So we find structural support, but we don't find Cooksonia extant anymore. So this is what we think was the primitive first system for transporting water. The next evolutionary advance was to make a secondary cell wall, which is now composed of lignin. So the first primary wall is cellulose, the second wall on the interior is lignin, and then to have perforations or pits at the end of a cell so the water could be held and moved up under tension in one cell and then transferred up to the next cell through these pit membrane connections. They were closed at the end with these perforations, and these cells are called tracheids. So tracheids are characterized as being thin and by having a pitted structure at the end, but not open tubes. This rigidity allows a plant to sustain a vertical position. It also allows the water to be moved up under tension. It's quite an effective system. Even today, in the low CO2 world, highly evaporative world that we have, we can find some conifers that reach a height of 100 meters, well over 350 feet. These would be the coastal redwoods and the giant sequoias of California. In Alaska, Washington, Oregon, and those wetter regions, we will find Douglas fir that reach a height of about 75 meters or about 250 feet. In our part of the world, most of the trees are not much higher than about 40 meters or about 130 feet. Still quite high. So what we have is water being pulled up from the soil all the way up to the top evaporating tissues while held under tension. If you cut a tree stem in half, what you'll find are a series of concentric rings. The concentric rings represent the production of tracheids, which happens each and every year. When cells are dividing rapidly, they have a thin cell wall. When they're dividing slowly and they're being reinforced with lignin, then they're smaller. So if we begin to look at this cross-section here, the lighter colored portion represents the growth in terms of cell division of xylem early in the season. By late summer, when water is less present and the conditions are more stressful, the cells don't expand as much. They still divide, they still expand, but not as much. That results in much smaller rings, much smaller cells, and that's this darker appearance. So what you're looking at here is summer growth, spring growth, summer growth, spring growth, summer growth, spring growth. And the magnitude of the distance in between rings represents an indication of how much growth that took place that year. And people have been able to correlate the width of a tree ring with precipitation and other climatic factors. And this is one way in which scientists begin to reconstruct the history of wet periods and drought. Since we know conifers can live for a long time, I mentioned that the bristlecone pine can live for about 5,000 years, we can reconstruct wet and dry periods with an almost annual degree of confidence.